Hi, and welcome back to my channel. In today's video, we will be taking a look at muscle tissue, looking at the different types of muscle tissue, the functions of muscles, properties of muscles, taking a closer look at the connective tissues found in muscle. We'll be looking at the skeletal muscle fiber, the components of that, also the neuromuscular junction, and how a muscle contracts. If you enjoy this video and its contents, I ask that you like, share, subscribe, and turn on notifications so that you never miss out on a new video. In your class, you should have already looked at the bones of the skeleton and the joints between them, so it's logical that we next consider the muscles, which move the bones, and then eventually talk about the nerves that control them. However, muscle does many other things besides moving bones. It is a contractile tissue that's divided on its histological structures into three types. The three types of muscle include skeletal muscle, cardiac muscle, and smooth muscle. The main focus of this video will be skeletal muscle. However, on the next slide, we'll take a closer look at these three muscles, where they're found, and what they look like. This table nicely depicts the difference between skeletal muscle, cardiac muscle, and smooth muscle. In the first column, you'll see skeletal muscle. Skeletal muscle, just like its name implies, is attached to the skeletal system. It is used so that we can control the movement of bones in the skeleton. This muscle is voluntary, so we decide when we're going to move, unless under certain circumstances. Cardiac muscle is an involuntary muscle, which means we don't have to think about it moving. This muscle is found in the walls of the heart and is used for contraction so that blood can move through the heart. Smooth muscle is found in the organs, in the walls of the organs, and this muscle is used for organ movement. This muscle is also has an involuntary muscle movement. So if you look at the pictures below each one, each of these pictures down here show what these look like underneath the microscope. What you'll notice is that skeletal and cardiac muscle both have striations throughout the muscle. Skeletal and cardiac muscle are very similar in how they work, though there are some differences. In smooth muscle, though, you do not see those striations, and that's where smooth muscle gets its name. The contraction of smooth muscle is different than the contraction of both cardiac and skeletal muscle. And then the final picture on the bottom shows what it looks like with an artistic depiction, so you can see a little bit more. Some more differences is that skeletal muscle has a very long cells. They're made of very long cells, and they're multinucleated, so they have many nuclei in there. You can see them all around. Cardiac muscle branches, so you can kind of see this branching that you see down here. It branches off, and then it has these really dark lines right here, which is very characteristic of cardiac muscle. These are called intercalated discs, and we find these in between different cardiac muscle cells. And in the picture above it, you can also see the intercalated discs here nicely. So that's something that you only see in cardiac muscle. You don't see that in skeletal muscle. Cardiac muscle, unlike skeletal muscle, is uninucleate, okay? And so they're both uninucleate. Cardiac muscle and skeletal muscle both have striations, so that's really not distinguishing characteristics between them. Distinguishing characteristics are the fact that skeletal muscle is multinucleated, while cardiac muscle is uninucleated, and there are intercalated discs that are found in cardiac muscle that are not found in skeletal muscle. Smooth muscle, on the other hand, again, is smooth. All right, it's also uninucleate, though the way that it looks, it's very difficult sometimes to even see the nucleus in there. There's one. And you don't see striations, okay? So a very different looking muscle. You can see the muscle cell also on the bottom here, and that muscle cell, they kind of just stack on top of one another. So a very different makeup than we see with skeletal and cardiac. And that's because in organs, we have a very different movement of muscle than we have with skeletal and cardiac. Skeletal and cardiac produce these contractions, right, to move bone if we're talking about skeletal muscle, or to contract the heart if we're talking about cardiac muscle. But smooth muscle has more of a peristaltic contraction, a wave-like smooth contraction, mostly in the digestive system to help food flow through it. Okay, so different kinds of contraction found in each one. Let's talk about the functions of muscles. One of the functions of muscles is movement. Okay, with skeletal muscle, we're going to be able to have that movement to move from place to place, move parts of the body, breathing, circulation of the blood, if we're talking about cardiac muscle, feeding and digestion, that's talking more about the smooth muscle, defecation, urination, childbirth, even communication, the fact that you can move your mouth, form speech, to write. All of these types of communication are going to be assisted by the use of various muscles. Another function of muscles is stability. The muscles have the ability to maintain posture by preventing unwanted movements. There are also anti-gravity muscles that help to resist the pull of gravity and prevent us from falling or slumping over. And muscles also help to stabilize joints. Muscles can also control opening and passageways. We see this when things are moving through the body. There are muscles that form what are called sphincters. These sphincters usually have a voluntary skeletal muscle and an involuntary smooth muscle associated with them, just depending on where they are. That can vary, but sphincters can control openings and passageways. We talk about a few of these sphincters in the digestive system, but these are internal muscular rings and they help to control the movement of food, bile, blood, and other materials. We see these throughout the body. Finally, we also have heat production by the skeletal muscles. When the muscles shiver, such as when we are cold, this can create as much as 85% of our body heat. So when we are cold and our muscles start to contract and create that shivering, the energy that's used for that allows for heat to be created in order to warm us up, acting as a negative feedback system. Okay, so let's move on to the properties of muscle. One of the properties of muscle is electrical excitability, and this is the ability that muscle cells have that they can respond to certain stimuli by producing electrical signals called action potentials. And action potentials are pretty exciting, and we'll be talking about those later on. This is very unique to muscle tissue because besides nervous tissue, other cells just can't respond to this, and they cannot produce action potentials. So we'll be talking about the ability of muscle tissue to create an action potential. Another property of muscle is contractility, and this is the ability that muscle tissue has to shorten forcefully when it's stimulated by an action potential. And then, of course, when the action potential is over, it can relax. So that muscle can contract and shorten when it's stimulated. Another property of muscle is its extensibility. This is the ability that muscle has to stretch within limits without being damaged. We see this in all muscle types, but smooth muscle usually can have the greatest amount of stretching when we see things like eating a meal and the stomach stretching and expanding, the intestines being able to be stretched. So smooth muscle has a little bit more extensibility than the other muscles, but they all can extend to an extent. And then finally, there's elasticity. And this is the ability of the muscle tissue to return to its original length and shape after contraction or extension. Later on, as we talk about the structure of the muscle fiber, you'll see how muscle has this ability to contract, have that elasticity, and then go back to normal and be stretched. And so we'll take a look at those different things later on. Muscle tissue has 3 different layers of connective tissue that's associated with it. I'm going to go ahead and talk about them here, but then in the following slides show you pictures so that you better understand where these are found. So the first type of connective tissue is referred to as the endomysium. This is a thin sleeve of loose connective tissue, and it surrounds each individual muscle fiber. This also allows room for capillaries and nerve fibers to be able to reach the different muscle fibers. And this is going to be important when we talk about contraction and being able for the entire muscle to all contract at one time. The next layer of connective tissue is referred to as the paramecium. This is a slightly thicker layer of dense irregular connective tissue, and this layer of connective tissue surrounds the fascicles of muscle. The fascicles of muscle are bundles of muscle fibers, and they are then wrapped in paramecium. In this area, we'll see larger nerves and blood vessels, as well as stretch receptors. And then finally, we have the epimecium. This is a fibrous sheath of dense irregular connective tissue that surrounds the entire muscle. All three layers of connective tissue are found within the muscle, but they all then extend together beyond the muscle fibers to form a rope-like tendon, and the tendon is used to attach that muscle to bone. With the tendon then attached to the bone, when a muscle contraction occurs, then we can have movement of the bone. The other type of connective tissue that we see in the muscle is called the fascia. This is a sheet of dense connective tissue that supports and surrounds muscles and other organs of the body. This holds together muscles with similar functions, and we see it again surrounding the entire muscle. The fascia also allows free movements of muscles, carries nerves, blood vessels, and lymphatic vessels, and it fills the spaces between muscles. This image shows the different layers of connective tissue and where they would be found. So let's kind of go first deep within the tissue and look at each individual muscle cell. So right here, okay, you can see that right here we find a skeletal muscle fiber or a muscle cell. And so skeletal muscle cells are also referred to as skeletal muscle fibers. And if you remember back at the beginning of this lecture, muscle cells are very long. They're very long, they're multinucleated, and so they're also referred to as fibers. An individual muscle fiber or muscle cell is surrounded by an endomysium. So surrounding this cell would be endomysium, and that's what they're showing you in this picture. We would see endomysium that surrounds all these individual cells. If we blow this up so you can look inside a little bit better, you'll see that there are individual muscle fibers in this area here. I'm showing you them, and then surrounding them is an endomysium, okay? So that connective tissue that surrounds each muscle fiber is the endomysium. A bundle of muscle fibers that you see here is referred to as a fascicle, okay? So as we get larger, this whole thing would be referred to as a fascicle, and then that entire fascicle would be surrounded by what we call paramecium, okay? And you can see that again in the picture over here that I just referred to. We have endomysium surrounding each of the individual cells, and then all surrounding the muscle fascicle, we have the paramecium that surrounds that. So then if we look at the entire muscle, which is here, the entire muscle would have several fascicles within it. So each one of these would be a fascicle. If we were to look into each fascicle, we would have individual muscle fibers. And so the entire muscle is composed of all these muscle fascicles, and it is surrounded by an epimecium, okay? So the epimecium surrounds the entire muscle. The paramecium surrounds each fascicle, and the endomysium would surround each muscle cell. You can also see the tendon that extends from the muscle. Remember, all three of these connective tissues extend out towards the back, and they all come together to form that tendon that would then attach to bone. So it's very interconnected here. The one thing that they're not showing you is the fascia, and the fascia would be surrounding the entire muscle. So it would be over the epimysium, and it would be surrounding the entire muscle. This figure shows the organization of a fascicle, so you can see very close up what a fascicle looks like. Remember, a fascicle is a bundle of muscle cells or muscle fibers. So each one of these is an individual muscle cell. They're surrounded by an endomysium. Each individual muscle cell is surrounded by that endomysium. If we take a look inside the muscle cell, we can see that there's a nucleus. They have several nuclei throughout. Mitochondria. Remember, muscle cells are a specialized cell in order to be contractile and have that contractility. And for that to happen, a lot of ATP is needed. And in order to make all that ATP, skeletal muscle cells and all muscle cells have a great abundance of mitochondria in them. So these cells have more mitochondria than normal cells. And so you see that in there to make that ATP. So each individual muscle fiber is surrounded by the endomysium. All the muscle fibers together to make a fascicle is then surrounded by that perimysium. So that's a little bit of a close-up picture so hopefully you get an idea of what you're looking at. If we take it even further and look at connective tissue in a muscle cell underneath the microscope, so now we're looking at a slice of skeletal muscle. We can see how it's set up in the same way, okay? Here they have the endomysium circled, so each one of these little areas in here is a muscle cell itself or a muscle fiber. Remember, this is cross section, so we're looking down at it, and then the space surrounding it would be the endomysium in there. Each bundle of these would be the fascicles, so bundle of muscle cells, and you can see that they've circled that on this side over here, and surrounding them would be the perimysium. Okay, so connective tissue surrounding each area, and then finally surrounding the entire muscle, we would have the epimysium. So another look at this underneath the microscope so you can see how this looks. So now we're going to take a closer look at the muscle fiber itself. And I know that all of these concepts can get very confusing because of the way that muscle is set up. It looks very similar even as you go into the deeper layers. So what you're looking at now is one muscle cell, one muscle fiber, not the perimysium like we looked in the slide before. You're looking at one muscle cell. So we're going to look deeper at this and see how it's set up, and then eventually how we can have a contraction in the muscle. So one thing you'll note that's different than the picture before is now the outer layer is the sarcolemma, and the sarcolemma is the name for the plasma membrane in a muscle fiber. The next thing that you'll notice that's named similarly is the sarcoplasm, and the sarcoplasm is the cytoplasm of the muscle cell. Again, a specialized name just like the sarcolemma so that we immediately know that we're referring to a muscle cell. So sarcolemma is the plasma membrane, sarcoplasm is the cytoplasm. In the sarcoplasm of these specialized cells, we find a large amount of glycogen, and this is needed for the contractions that the muscle cells do. Another area for you to take note of are these areas that are here in blue. These are called the transverse or the T-tubules. We refer to these areas a lot when we talk about action potentials and the ability of the signal to pass throughout the entire muscle cell. So what you'll notice is that within the muscle cell itself, these are all throughout the muscle cell, and this allows the signal to travel very quickly through the muscle cell. What signal? We'll talk about that idea later on. But for now, just kind of see how that's made up. Right next to the T-tubules, you'll notice that there's this area here. This area over on this side and all next to the T-tubules is referred to as the sarcoplasmic reticulum. This is the endoplasmic reticulum in these cells, and it's very, very specialized in this area because this is where calcium release is going to come from. And we'll talk about that later on. If we continue to take a closer look at the muscle cell, we'll notice that the muscle cell is made up of myofibrils, which are these areas here. Myofibrils are bundles of filaments that fill the sarcoplasm of a muscle cell. And if we look closer into an individual myofibril, you'll see that the myofibril is made up of myofilaments. And those myofilaments are thick and thin. Okay, so we have a thicker looking one and a thinner looking one. And this is going to be, our thicker one is myosin and our thin one is actin. And we're going to be talking a lot about these myofilaments because the movement of these sliding across one another, and we'll refer to how that happens later on, is very important because that's how a muscle contracts. So just so that you can kind of get a look at this as a whole, each one of these myofibrils is composed of these different myofilaments, and they've shown you here what this looks like. And so this is throughout just one muscle cell. And remember, one muscle cell then bundles into a fascicle, so a bundle of muscle cells, and then all those bundles of muscle cells eventually makes an entire muscle. So we go all the way down to the nitty gritty. This is what it looks like. And so when we talk about contraction and talk about what's happening at this microscopic level, you can put that all together as a whole, and all the myofilaments, or most of the myofilaments of the muscle cells, doing this in conjunction throughout the entire muscle to have a contraction, okay? But in order for you to kind of understand what's going on, we have to take it down to the microscopic level because this is where the action is going to be happening, all right? In the next slide, we're going to take a look even further at a very simplistic version of a muscle fiber. The figure that you're looking at now is a very simplistic view of a muscle fiber. This is so that you can hopefully get a better understanding of how a muscle cell is set up, so that when we talk about how a muscle cell contracts, you can understand the physiology of that. So again, up on top here, we'll talk about this area right here. This is our transverse tubule, or our T-tubules. They were blue in the figure before. And these again are invaginations of the plasma membrane that creates these tunnels throughout the muscle fiber. And so you see that here. And next to the T-tubules, we see that there's the sarcoplasmic reticulum. The sarcoplasmic reticulum is really important because these are reservoirs for calcium. So you can see calcium in them. All right, and so this is going to be responsible for the release and the uptake of calcium in order for a muscle contraction to occur. We also see that the myofibrils are made up of the myofilaments. Again, you see the thick and thin filaments. So you see the myosin and the actin, okay, in there. And attached to each myofibril, you also see some other proteins, okay, the dystrophin and the membrane protein that are connecting it to the cell itself. So it's connected all to the sarcolemma or the plasma membrane. So these are not free floating, they are attached, and so that can allow for movement of the entire cell by being attached to the plasma membrane. Some other important things that we see within the cell are the mitochondria, okay? A lot of mitochondria, like I mentioned before, because we need a lot of ATP for the muscle cell to contract. There's also myoglobin, which has a higher affinity for oxygen than hemoglobin, so we can have a little bit of oxygen storage. And then glycogen granules, which can be broken down and again used for energy. So we see these are all special things that we see in these specialized cells. Here we can take a closer look at the myofibril itself. So I'm going to point out some things so that you can understand some terminology before we talk about muscle contraction. In this area right here, we refer to this area as a Z-disc. So you'll see a Z-disc here, you'll see a Z-disc here, and the entire area from Z-disc to Z disc is referred to as a sarcomere. And the sarcomere is called the basic functional unit of a myofibril. But a myofibril has several of these, so they'll line up all one next to the other, next to the other. This image is just kind of focusing on the one so that we can blow it up and look at it in more detail. But know that there's not just one sarcomere per myofibril, there are several. So they would line up all one next to the other. But from one Z disc to one Z disc, we call this whole unit a sarcomere. The next thing that we're going to look at, which you've seen again and again, is that this sarcomere has overlapping thick and thin filaments, okay? Remember, the thick filaments are the myosins, and the thin filaments are the actin. And what you'll notice is that the thick filaments and the thin filaments overlap, okay? Again, that's going to be important for how that muscle contracts. In this picture down here, you can see in a little bit more detail. So here's the thick filament and the thin filaments, and they overlap one another. Another important protein to take note of is this protein here called titin, which you'll see it right here. It is a huge springy protein. It's a key structural protein, and you'll notice that it flanks each thick filament. So the myosin, it flanks each thick filament to the Z disc. So everything that we're looking at in the muscle cell is all connected to one another in some way. So this titin protein is responsible for anchoring this thick filament to these Z discs. It helps to stabilize that thick filament and also center the thick filament between the thin filaments or the actin. It also works to prevent overstretching. A few other things to notice. You'll notice that they label this area right in here. You see this line right here. They label this area the I band, okay, which would be in here. And the I band is light. It's a less dense area. And in this area, when the muscle is relaxed, we have thin but no thick filaments. The other band that they label is the A band, okay? And the A band, which you see in this area right here, the A band is darker and it's part of the sarcomere where you have basically the length of the thick filament. So in the A band, you have thick and thin filaments within the A band. In the I band, it kind of goes from sarcomere to sarcomere, and you only see the thin filaments. So not only do you see striations between the thick and the thin, okay, but you'll also see striations going from I band to A band because you'll have a lighter area in the I band, a darker area in the A band. Now, as you can imagine, when a muscle contracts, it's going to change a little bit what this I band and A band look like, and so that's why we've given them that nomenclature. The H zone is the narrow center of each A band where you only see the thick filaments, and so this would be only thick filaments, only myosin would be found here, and no thin filaments. And finally, right in the middle of the sarcomere, we find what is referred to as the M line. This is where the thick filaments or the myosin attach right here in the center. This figure is an electron micrograph where you can look into the cell and see these different areas. So again, from Z disc to Z disc is what we refer to as a sarcomere. You can see in this picture there are several. They line up one next to each other in a muscle fiber. The I band is lighter than the A band. And so we can see striations of I band and A band, and we can also see striations as thin and thick filaments overlap one another. Here's a nice picture of mitochondria. There are several of them within skeletal muscle cells, and we also see the transverse tubule and the sarcoplasmic reticulum that will be in here. Taking a closer look at the thick filaments, remember again, these are referred to as myosin. These filaments are made of a head and a tail. The head is shaped like a golf club. There are two chains that are intertwined to one another, and they eventually form the tail of the myosin. It's a double globular head, and the head directs outward in a helical array throughout the bundle. So you can see how they all kind of face outward in this bundle. And in the middle, there's a bare zone, and this bare zone has no heads in the myosin. The actin itself is referred to as the thin filament, okay? And sometimes it's referred to as a string of beads. You can see they look very bead-like right here. Actin is also associated with two other molecules, tropomyosin and troponin, okay? And these are all very important in how the muscle is going to contract. So a couple things I'm going to point out. The actin, which are right here, these bead-like structures. In the actin, there are these little areas, and you'll see those areas are referred to as the myosin binding site. And this is where myosin is going to bind in order for these filaments to slide over one another. And so that's where the myosin is going to bind. But what you'll notice in its normal position, these sites are blocked by tropomyosin. So that's what this molecule is doing right here. It is blocking the myosin binding site on the actin. And troponin is attached to the tropomyosin. Now, troponin is important because troponin is where calcium will bind. And if calcium binds to troponin, it's going to change the configuration so that tropomyosin will move and expose these myosin binding sites. So normally, without calcium present, this is what it's going to look like. You'll have your actin, you'll have your tropomyosin binding, you'll have your tropomyosin blocking the myosin binding sites on that actin, and then the troponin is attached to the tropomyosin. So all of these are very important in how this works. When talking about how a muscle contracts, we can separate the proteins into two different types. We have the contractile proteins, actin and myosin, those thin and thick filaments. They're the ones that do the work, which is why they are called the contractile proteins. And then we have the regulatory proteins I just mentioned, tropomyosin and troponin. These are important because they can act like a switch and determine when the fiber can and cannot contract, depending on whether it's blocking the myosin binding sites or not. So troponin, again, is going to be responsible for changing its shape a little bit, moving tropomyosin off moving tropomyosin off actin and exposing those myosin binding sites. Now that you hopefully understand how a muscle cell is set up on the inside, let's go back and look at the neuromuscular junction where a neuron is going to connect with a muscle cell. So this picture here shows you a muscle cell fiber or a muscle cell. This is the sarcolemma or the plasma membrane of that muscle cell. And in this area above, you'll see the axon of a motor neuron. So the motor neuron is going to extend out, and the axon of the motor neuron is going to eventually end in this area that's referred to as several names. Here they're referring to it as a synaptic end bulb. You may also have seen it referred to as a bouton, but the end of the axon has this kind of outer swelling area, and this is going to meet up with the muscle cell. And basically this is where the action is going to take place in order for a contraction to occur. Here's another depiction underneath the microscope. This is a specialized microscope called a scanning electron microscope. It's capable of taking these 3D pictures. So really cool picture of what this looks like. So here we have the neuron, the axon of the neuron extending out, and then again you can see where the axon is going to have the end terminal, and then coming out from that end terminal is going to be those synaptic bulb ends. So they're all over the place, and that's where the neuromuscular junction is going to happen. So where the neuron meets the muscle, specifically where the synaptic end bulb or the bouton of the axon meets the muscle cell sarcolemma, that is what's going to be referred to as the neuromuscular junction. Now, they don't actually touch, even though they look like they're touching. There is a little bit of area between the two, but imagine now all over the muscle there are several neurons, several synaptic end bulbs, and so this is going to be all over the muscle cell. And so here's a close-up depiction of this so that you can understand this better with this artistic depiction. Again, here we have the axon terminal coming down. It's going to end in this outswelling right here, which is referred to as the synaptic end bulb, and it's going to meet up with the sarcolemma of the muscle cell. So this area here where they both meet is referred to as the neuromuscular junction. I mentioned that they don't really touch. There's this area right between them called the synaptic cleft, where there's an area so that we can have movement between the two. And you'll notice there's a lot of things going on between the two. This is how they can communicate with one another, so that if the neuron itself has an action potential going through it, it's going to end at the synaptic end bulb. Things are going to happen, which we're going to talk about, and then that can have an effect on the muscle cell in order for the muscle cell to have a contraction. I've shown this picture one more time just so that you can get it back in your memory. Once we move from that synaptic cleft area of the neuromuscular junction and things happen there, then the signal can travel through the T-tubules. Remember, these are invaginations of the sarcolemma. The signal can travel through these areas and then have effects on the sarcoplasmic reticulum that is sitting right next to it, and then eventually having that effect on the actin and myosin, okay? I know I haven't specifically told you what's happening yet, but I want you to get a picture in your mind of how this is all set up so that when we do start to talk about this action potential and things happening here, you will understand that better. I really like this figure because we're going to be talking about muscle contraction and how that muscle contraction occurs. And this figure is a really good representation of what's happening with these myofilaments. So we know that actin and myosin are situated over one another here. So we have the myosin, the thick filaments, the actin. And in a relaxed muscle, this is what it's going to look like. And you can see the picture on an electron micrograph right here of what that looks like. So we have our I band, that's the lighter area, our A band, that's the darker area. And what happens is, as the muscle starts to contract, the actin and the myosin start to slide over one another. And again, we'll talk about how that happens. But what you'll notice is that there's going to be a changing of what the I band and the A band looks like to eventually where we can maximally contract a muscle. You can see how the I bands get much smaller in relation to the A bands as these proteins start to slide over one another here. So this is a nice representation of what's happening. The muscle is going to contract. These proteins are going to slide over one another, causing the darkening that we're seeing here compared with the first one, and then they'll relax back to the beginning. So how exactly does a muscle contract? In order for a muscle to contract, we have to have a series of events that occur. The first thing is, if we're looking at our neuromuscular junction, so just a reminder, this is the synaptic bulb that is here at this end. This is the end portion of the axon. So the axon of the neuron would be above here, and there are several of these end terminals that come off an axon. And they come and they meet with the muscle. So this down here would be the muscle. This would be the sarcolemma of the muscle or the muscle cell membrane. And up here we have that synaptic bulb of the axon, or can be referred to as the bouton. So here they are meeting in this neuromuscular junction. And so we have to have a series of events that occur in order for the muscle to contract. And the first one is that the neuron has to have an action potential that runs through it. Now later on, when we talk about neurons, and we talk more specifically about neurons, then in that chapter, we'll go into more detail as to how a neuron has an action potential, because it's a little different than a muscle. But for right now, understand that an action potential does travel through the neuron here. And when that action potential travels through the neuron, it's going to change the voltage of the membrane, okay? Now, neurons and muscle cells at rest are normally very negative. Their resting membrane potential is very negative, and that can range anywhere from minus 90 millivolts, which you would see more in the muscle cell, to minus 70 that you would see more in the neurons. And so the resting membrane potential of these cells is very negative. And what happens is a change occurs in there that allows this resting membrane potential to shift. We're more going to specifically talk about how the muscle cell membrane shifts. But this action potential is going to course through the neuron, and it's going to end up in this synaptic bulb portion, okay, this end bulb portion, or the Bouton portion of this. And what's going to happen is that's going to cause the membrane potential to shift from something very negative to something more positive. And these channels right here are called calcium voltage gated channels. And as that membrane potential shifts in that Bouton area, in that synaptic bulb area, that's going to cause these voltage gated calcium channels to open. So normally they're closed at rest, and as we change that shift from negative to being more positive, they're going to open. Now remember, again, that shift is happening because an action potential is running through that neuron. And once these channels open, that's going to allow for calcium that is sitting outside to move inside, okay? So calcium is going to come in to this area through these channels. There's a lot of these channels, so calcium can flood in from the outside. It's normally outside. It's going to come in to the inside, and this calcium is going to cause these synaptic vesicles to release their contents. So what I've drawn here in the bottom portion, these areas right here are referred to as synaptic vesicles. And in these specific synaptic vesicles, we have a neurotransmitter called acetylcholine. Now, different neurons can have different neurotransmitters, but for our purposes right now, talking about skeletal muscle contraction, we are going to refer to acetylcholine being in this area. So when the calcium comes in, it encourages these synaptic vesicles that are all sitting here ready to be released to fuse with the plasma membrane of the synaptic bulb and then release its contents into the synaptic cleft, okay? And so here's the synaptic cleft, this area that's I'm drawing these dots in. And now acetylcholine is going to be released. So imagine calcium floods in, and it's going to cause all these vesicles that are sitting here to come fuse with the membrane and release the acetylcholine into the synaptic cleft. So now acetylcholine's in the synaptic cleft, and we have another set of ion channels here. Okay, so here's some more ion channels, but instead of being voltage gated channels, these channels are ligand gated channels, and the ligand that binds to these channels is acetylcholine. So these ligand-gated channels respond to a ligand binding them in order for them to open, rather than a voltage change. So in the neuron, we saw a voltage change to allow calcium in. In this case, these ligand-gated channels are going to respond to acetylcholine binding. So acetylcholine can then bind, now that it's in the synaptic cleft, it can bind to these ion channels, and the binding of the acetylcholine is going to now open up these ligand-gated channels. Once these channels open, this will allow the influx of sodium. So sodium's hanging out here too, outside, and so sodium can then come in through these open channels. Okay, so we're going to just have sodium as they open up. I know I didn't draw them all opening up, but the acetylcholine will be enough that they'll open and sodium will flood into this area. Now remember that the membrane has those T tubules, okay? So the sodium is actually going to travel through the T tubules. And sodium is positive, and I just mentioned to you before that the resting membrane potential of skeletal muscle cells is very negative. And so as we allow this positive ion to come in, it's going to change this very negative number, and it's going to move to become more positive, okay? And this is going to have an effect as it flows through. So now sodium is coming in and it's traveling through those T tubules. The fact that as sodium travels into the cell, causing the resting membrane potential of the cell to become more positive, this is referred to as depolarization. So the cell is depolarizing. The membrane of the cell is depolarizing, okay, as that sodium comes in. And that sodium, like I just said, travels through the T tubules. And remember that the T tubules sit right next to the sarcoplasmic reticulum. So as that changes, it's going to start to change the membrane potential right next to the sarcoplasmic reticulum. And so then that positive signal is going to swoop through the T tubules, depolarizing the cell as it goes through the cell membrane, and it's going to cause the release of calcium from that sarcoplasmic reticulum. So if you remember before, this is what I've drawn. I've drawn the T tubules here. So these are the T tubules. And then in this area here is the sarcoplasmic reticulum that sits right by it. So as the sodium travels through the cell and depolarizes the cell membrane, it's going to cause the release of calcium from these T tubules. So calcium is going to be released into the cell from here. And the reason that it gets released is because there are voltage gated calcium channels on the sarcoplasmic reticulum. And I didn't draw them here, but we can just draw a few of them, some channels on here, okay? And as the membrane potential changes, as the sodium travels through the T tubules and changes the membrane potential, it's going to cause these voltage gated channels to open, because that membrane potential will change as well. And when the voltage gated calcium channels open on the sarcoplasmic reticulum, this is what is going to allow the calcium to come out, okay? And calcium is really important because that's what's going to be needed in the next step. So now we're in the cytoplasm of the cell, where we're going to find those myofilaments, okay? So remember, we have actin and myosin, and they overlap each other, and we had talked about them before. And now we have calcium, so calcium's in the cell too. And actin has tropomyosin and troponin that's attached to it, okay? So remember, this was our thin filament, and this was our thick filament. And what's going to happen next is that calcium is going to bind to troponin. And I had mentioned that before. And calcium is going to bind to troponin. That slightly changes the shape of troponin, causing the troponin-tropomyosin complex to move away. Remember, troponin sits on tropomyosin, and this is going to cause this complex to move away. What is it moving away from? Remember, it's blocking, it's at rest, it's blocking the myosin binding sites on actin, but now that it moves away, it's going to open up the myosin binding sites on actin. Once those myosin binding sites on actin are open, now myosin can bind to actin, causing the sliding of the filaments over one another and allowing for a muscle contraction to occur. Now, this can only happen in the presence of calcium, so all the steps that we just mentioned have to happen before this occurs. So let's look more closely as to how actin and myosin filaments slide over one another. In this figure, we can take a closer look as to how actin and myosin slide over one another. So this is also referred to as the contraction cycle. And before this contraction cycle can start, calcium has already been released into the sarcoplasm of the cell. It has bound to troponin, and it has caused the moving of the troponin-tropomyosin complex. So now you can see that on the actin, the myosin binding sites are now exposed. Now you'll also find out why it's so important for the muscle cell to be able to have ATP. So the first thing that happens is that the myosin itself, the myosin head, has an ATP binding site. And myosin, the head of the myosin is going to split that ATP using ATPase to split ATP into ADP and a phosphate. So that's what you're seeing here first. So ATP first becomes split, and we get an ADP and a phosphate. When this happens, the splitting of that ATP reorients the head and energizes it for the next step. So in the next step now, once the ATP gets split by the ATPase, now the myosin head can bind to the actin. And so that's what the next step is going to occur. So now that the myosin binding sites are open and myosin has split the ATP, it can now bind to the myosin binding sites on the actin. The next thing that's going to happen is that the myosin heads are going to rotate towards the center of the sarcomere. So it depends on which side of the M line we're talking about, but they are going to both rotate toward the center of the sarcomere. And when this happens, this is referred to as the power stroke. Also during this step, you will see that ADP gets released. So during this power stroke step, ADP will be released. In order for the myosin head to detach from the actin, it has to bind another ATP. So ADP gets released during the power stroke, and then ATP will bind to the myosin head. This causes the release of myosin from actin, and then this can continue, okay? This will continue again and again as long as there's calcium present and as long as there's enough ATP to continue binding. In order for the muscle contraction to stop and the muscle to go into its relaxation period, there are a couple of things that need to happen. One, the signal from the neuron needs to stop. Once the release of acetylcholine stops coming from the neuron itself, then it can be rapidly broken down by acetylcholinesterase, which is an enzyme that breaks down the acetylcholine in the synaptic cleft. Acetylcholinesterase is again found in the synaptic cleft. If it wasn't there, then it wouldn't be able to break down the acetylcholine that had been released, and it would take a long time for the muscle to stop contracting. But because it is there, it can easily break down the acetylcholine that is there, and it breaks acetylcholine into acetate and choline. The acetate gets swept off by the synaptic cleft into the interstitial fluid, but the choline gets taken back up by the presynaptic neuron to be reused. The other thing that needs to happen in order for the muscle to relax is that the calcium channels in the sarcoplasmic reticulum need to close, and calcium has to be pumped back into the sarcoplasmic reticulum. In order for the calcium to be pumped back into the sarcoplasmic reticulum, it will require active transport. And so this is where another use of ATP comes into play. So as you can see, the myosin heads each require ATP, and in order to pump calcium back into the sarcoplasmic reticulum, that is active transport and requires ATP as well. So a lot of ATP is necessary for these things to occur. Once we get the acetylcholine released to stop, acetylcholinesterase to break down any remaining acetylcholine, and the calcium back into the sarcoplasmic reticulum, then the muscle contraction stops, and then the muscle can go through relaxation. So now that we've looked at how a muscle contracts at the molecular level, how can we get the entire muscle to contract all at the same time? In order to be able to do this, we can look at a motor unit. A motor unit is one nerve fiber and all the muscle fibers that are innervated by it. A single motor unit makes contact with an average of 150 muscle fibers within a muscle, and all of the fibers in one muscle unit can then contract within unison. So if it is innervating all of these muscle fibers and the signal comes from that axon of that neuron and it innervates about 150 of these different fibers, we can get a contraction of them all together because the signal is going to be transversed through all of them at the same time. In order to have an effective contraction, it is usually going to require the contraction of several motor units at once. We can divide motor units into small motor units and large motor units. Small motor units would be those for a fine degree of control. So there would be about 10 to 20 muscle fibers per neuron, and this is for very fine control, such as in your eye and hand muscles. Whereas large motor units is used more for strength than control, and very powerful contractions. So you can have a lot of fibers that are innervated by the neuron. In the gastrocnemius, you would see 2,000 to 3,000 muscle fibers per neuron. So there's a lot of them innervating. This is for a very powerful contraction versus a very fine-tuned contraction. So the large motor units are going to have more strength than control, while the small motor units are going to be used for the fine degree of control. Here's just an example of what that motor unit looks like. So we have the axons extending from the spinal cord down to the muscle, and you can see here's a motor unit innervating many different muscle cells, and another motor unit innervating many different muscle cells, okay? They're going to be getting the signal at the same time. The signal will travel through the axon, depolarizing the axon. Remember the synaptic cleft, we're going to have acetylcholine released from the neuron itself, and that's going to allow for sodium to rush into the muscle cell, depolarizing the muscle cell, and eventually have that calcium release for that muscle contraction to occur. But since we have these motor units, we can have that signal sent to many different areas of the muscle at the same time and have the muscle contract as a whole. We want to have an effective contraction. We don't want one muscle cell contracting because we wouldn't even feel that effect. We want the entire unit to be contracting at the same time. On a myogram, we can record muscle contraction. So we can see the different stages of muscle contraction. And there are 3 different periods throughout that muscle contraction. The first one is going to be the latent period. In the latent period, there is a brief delay between application of the stimulus and the beginning of the contraction. In this period, the muscle action potential will sweep over the sarcolemma and calcium ions would be released from the sarcoplasmic reticulum. The next period would be the contraction period. This is where the calcium is going to bind to troponin. The myosin binding sites on actin are exposed because troponin moves the tropomyosin from the actin, exposing those sites. And then myosin will bind with the actin, and this is going to result in that power stroke that we had just talked about. During that power stroke, that's going to generate the force of the contraction. The final period would be the relaxation period. This is the period where calcium is actively transported back to the sarcoplasmic reticulum. Tropomyosin is going to cover the myosin binding sites on actin because calcium is going to be removed. and then the power stroke will cease and the muscle fiber can relax. Duration of these periods depends on the type of skeletal muscle. Fast twitch muscle is about 10 milliseconds, and slow twitch muscle would be closer to 100 milliseconds. This figure gives you an overall idea of the different periods of muscle contraction and the force of the contraction. So here we have the force of the contraction and the time graphed. In the latent period, this is where the muscle is getting ready for a contraction to occur. In the contraction period, the calcium has been released and the muscle is going through a contraction, so the force of contraction is increasing. And then as calcium goes back into the sarcoplasmic reticulum, we are going back through that relaxation period where the muscle is returning back to its resting state. This figure shows different myograms for different types of contractions. The first figure right here, A, single twitch, shows a twitch contraction, which is a brief contraction of all the muscle fibers in a motor unit in response to a single impulse in its motor neuron. So the motor neuron gets one impulse. We can see this in the action in the contraction that's occurring here. It's receiving one action potential. So the action potential here is coming from the neuron and it's resulting in a single twitch contraction. We can then move to wave summation. Wave summation is when the stimuli arrive in closer sequence, causing larger contractions. So what happens here, down here you can see there's an action potential and then there's another action potential. So it's receiving the signal from the neuron quite in close proximity with one another. So what's going to happen is the muscle is going to contract and then it's going to contract again. What's happening here is that the muscle is contracting. It's not able to go all the way back to its resting period. And so the contraction that comes afterwards is going to be much stronger. This is because not all the calcium went back. The calcium is still there, and that can result in a stronger contraction afterwards. The next one is unfused tetanus. This is where the muscle can partially relax between stimuli. So what's going to happen here is that the action potentials are coming pretty consistently, but they're coming with enough space in between them that the muscle can partially relax. So what happens is a contraction happens. You see in this area right here, it can partially relax before it contracts again, partially relax, and then it contracts again, and then partially relax before it contracts again. What you can also notice is that every contraction is stronger than the last because of the fact that it does not fully relax and calcium is still present. Now, this is something very specific to skeletal muscle. Cardiac muscle cannot undergo tetanus. Cardiac muscle has to relax, so this does not affect cardiac muscle. So cardiac muscle does not respond in the same way. This is very specific to skeletal muscle in that skeletal muscle can react in this way. And then finally, we have what is referred to as fused tetanus. And this is where the skeletal muscle fiber can't relax at all. So the signals are coming in such close proximity with one another. The action potentials are coming in such close proximity from the neuron itself that the muscle cell can't even relax. So what you see here is just a consistent, sustained contraction that is occurring because of this, okay? In these cases, cardiac muscle cannot do this. Only skeletal muscle can respond in this way until, of course, the muscle becomes fatigued and then it just can no longer contract anymore. So skeletal muscles vary in their myoglobin content. And remember before I had mentioned that myoglobin binds to oxygen with a higher affinity than hemoglobin. So muscle cells can have a little bit of extra storage of oxygen within them, which is going to be needed for the contraction. And depending on how much myoglobin they have, they can be labeled as either red muscle fibers or white muscle fibers. Red muscle fibers are going to be referred to as red because they have a higher myoglobin content and therefore have a darker appearance. They have more mitochondria and also more blood capillaries, where white muscle fibers have a lower myoglobin content and they appear pale. We can then divide these fibers more specifically, talking about slow oxidative fibers. These are the smallest in diameter. They are the least powerful type of skeletal muscle fiber. They appear dark red because they have large amounts of myoglobin and many blood vessels, so they are referred to as red muscle fibers as well. There is much mitochondria, and they have a slow speed of contraction. They are very resistant to fatigue because of these features and capable of a prolonged sustained contraction for many hours. These specific muscle fibers are adapted for maintaining posture and for aerobic endurance type activities such as running a marathon. Fast oxidative glycolytic fibers are intermediate fibers because they're intermediate in diameter. They have large amounts of myoglobin and many blood capillaries, so they are also dark red in appearance. They have a lot of ATP that they get by aerobic cellular respiration, and they can get some ATP by anaerobic glycolysis. They have a moderately high resistance to fatigue, and they have a faster speed of contraction than the slow oxidative fibers. These contribute to activities such as walking and sprinting. And lastly, the fast glycolytic fibers are the largest in diameter. They contain the most myofibrils and have the most powerful contractions. These fibers are white. They are low in myoglobin. They have few blood capillaries, large amounts of glycogen, and generate ATP mainly by glycolysis. They contract strongly and quickly. They are fast twitch fibers. Intense anaerobic movements of short duration are best for these. Examples are weightlifting or throwing a ball. These fibers are going to be able to fatigue quickly. The distribution and recruitment of fibers. Most muscles have a mixture of these different fiber types that we just talked about. The proportions can vary. This can depend on the action of the muscle, a person's training regime, genetic factors. For example, for posture muscles, you're going to have a high amount of slow oxidative fibers. For arms and lifting, you would have a high amount of fast glycolytic fibers. Your leg muscles that are used for supporting the body, walking and running, would have large amounts of slow oxidative and fast oxidative glycolytic fibers. Even though the skeletal muscle can vary in the content of the fibers, the skeletal muscle fibers of any given motor unit are all of the same type. This figure shows a transverse section of 3 types of skeletal muscle fibers, and you can see how they vary in their color depending on how much myoglobin you have. So your darkest one would be your slow oxidative fiber because it has the most myoglobin right in here. Your fast glycolytic fiber would be your whitest one. And then you can see the intermediate fiber, that fast oxidative glycolytic fiber, is a color kind of in between the two. So more myoglobin than the fast glycolytic, but less myoglobin than the slow oxidative. So let's talk a little bit about muscle metabolism. All muscle contraction depends on ATP. I've mentioned already many times that there is a lot of ATP that is needed in order for the muscle to contract. The muscle goes from being fairly inactive to being involved in a contraction and using a great deal of that ATP. For the muscle to contract, myosin needs that ATP to convert it to ADP, and then ATP needs to bind again. ATP is also needed in order for calcium to be pumped back into the sarcoplasmic reticulum. So the ATP that is present in the cells is enough to power contraction only for a few seconds. If the muscle is going to contract past that time, then the muscle fiber must make more ATP. So how can the muscle make more ATP if it's needed to constantly continue to contract? There are 3 ways that the muscle can make ATP. The first way that ATP can be created is from creatine phosphate. Another way is by anaerobic cellular respiration, and a third way to make ATP is from aerobic cellular respiration. We are going to talk about these three ways in the following slides. The first way that the muscle cell can make ATP is from creatine phosphate. Excess ATP that is in the muscle cell at rest is used to produce creatine phosphate. Creatine phosphate is 3 to 6 more times plentiful than ATP in the sarcoplasm of the cell. When contraction starts and ADP levels start to rise, right? So ADP levels start to rise as a contraction starts. Creatine phosphate is going to catalyze the transfer of a high energy phosphate group to ADP to form the new ATP molecules. This is the first source of energy when muscle contraction begins. So because there is so much creatine phosphate in the muscle cells already, as the muscle cell starts to need more ATP, this is going to be the first source that is going to be used. With this can go for about 15 seconds of contractions, which is enough energy for about a 100-meter dash. So very quick source to be able to get ATP very quickly. The next way the cell is going to be able to generate ATP is from anaerobic cellular respiration. So once we pass that 15-second mark, then the glucose that is within the cells is going to be catabolized to produce ATP. So I had mentioned before that there's a lot of glycogen in the muscle cells. We can break down glycogen into glucose, and then from glucose we can catabolize that to eventually produce that ATP. This is used during periods of heavy activity where there's not enough oxygen for pyruvic acid to enter into the mitochondria. So in this case, since pyruvic acid, there's not enough oxygen, then this is going to release lactic acid. So usually for about 30 to 40 seconds, for the first 15 seconds, we use the creatine phosphate. 30 to 40 seconds more, we're going to be able to use this anaerobic glycolysis. And then eventually enough oxygen will be in the muscle cells to be able to move to aerobic cellular respiration. Finally, in aerobic cellular respiration, this is where oxygen is needed in order to produce ATP. For each glucose, you are able to make 36 ATP by cellular respiration, okay? A very long, involved process, but we can get 36 ATP. So obviously, this is the best generation of ATP, and we can get the most, of course, from this. So through cellular respiration. Now, in order for this to happen, oxygen is needed, so this can last for minutes to hours, okay? So normally if a person were to start exercising and running, once those muscle cells run out of ATP, they're going to start to use the creatine phosphate method to generate that ATP for about 15 seconds. Then you're going to go into anaerobic respiration because there's not going to be enough oxygen. You went from resting to running, and now there's just not enough oxygen in the muscle cells to be able to continue to make ATP. So it's going to go through a small stage, 30 to 40 seconds of anaerobic respiration, where it can still make ATP, not as much as in aerobic respiration. And then eventually your breathing, your respiration rate, your heart rate will all catch up. And so you'll be able to have enough oxygen in the cells, and you can continue this on for a much longer period of time through aerobic respiration, creating that ATP that is needed for the muscle cells to contract. Once you are finished doing the activity that you are doing, the body is going to have to replace all the ATP that was used, all the glycogen that was used, and this is going to take some time where the body is going to be able to replenish those systems again in order to get ready for the next bout of exercise. I want to thank you for watching my video. I hope this helped you with muscle tissue and understanding muscle tissue and how the muscle contracts and works. If you have any questions, please don't hesitate to leave them in the comments below. Thank you.